next speaker, McLaughlin, from the Department of Microbiology and the Elementary Pharmabiotic Centre, APC for short.
My talk is Listeria monocytogenes and the battle for iron inside the body. So just a few facts about Listeria. Listeria is a bacteria and it's found most places in the environment, including soil, water, and vegetation. So Listeria is responsible for the disease listeriosis, which is caused by eating contaminated foods. So for healthy individuals, most of the time they would just experience flu-like symptoms, but Listeria poses the greatest threat to a group called the Yappies, which is the young, old, pregnant, and immunocompromised. And by immunocompromised, I just mean people with pre-existing illnesses. So for this group, the symptoms can be far more severe, including sepsis, which is an infection of the blood, meningitis, miscarriage, or even death. So recently, Listeria has become recognized as a public health problem. And just to put things into perspective, as you can see here on the left-hand side, these are all bacteria that are associated with foods. And the top three, Campylobacter, Salmonella, and E. coli, while they do cause a lot of illness, the fatality rates are all less than 0.1%. Whereas Listeria might cause less illness, the fatality rate can be up to 20%. So also in terms of food recall due to bacterial contamination, Australia and New Zealand reported that 41% is actually due to Listeria. So how Listeria moves from farm to fork to infection. So like I said before, Listeria is found most places in the environment, and a survey was actually done in southern Ireland over 16 farms. And samples were taken from things like milk, silage, soil, water, etc. And of the 298 samples taken, 57 were actually contaminated with Listeria. So that's a pretty high percentage. So when animals are infected with Listeria, that can lead to contaminated milk or meat. And food, like vegetables, can also get contaminated from Listeria from the soil. So from food, Listeria can enter our digestive tracts. And because Listeria is such a hardy organism, it can often overcome stresses that would encounter here. So once Listeria makes its way into your intestine, it can actually cross over the intestinal cells and get into your bloodstream. And from the bloodstream, Listeria has the possibility of causing an infection throughout your entire body. So iron is extremely important, and this importance was recognized as early as the 1800s, when this quote was from. So gold is for the mistress, silver for the maid, copper for the craftsman cunning at his trade, good, said the baron sitting in his hall, but iron, cold iron, is the master of them all. So iron is the most abundant element on Earth, and it's essential to nearly all living organisms. So it's involved in many biological processes, including DNA synthesis and oxygen transport. For instance, iron is associated with a protein called hemoglobin, which is in your blood, and that allows for oxygen transport throughout your entire body. So the levels of iron inside your body, as well as in bacteria, must be tightly controlled, because too much iron can be toxic, and too little iron can have an effect on growth and reproduction. So why is iron important? Iron is extremely important during infection. So for example, bacteria like Listeria require iron to cause infection, and the body, in order to prevent infection, tries to hold on to that iron by incorporating it into proteins. So the battle for iron begins. So like I said before, in the body, in order to make sure there's no free iron available to bacteria such as Listeria, the body incorporates this iron into proteins. So for example, when you have dinner, the protein transferrin is released into your gut, and it binds to iron and transports that iron to your cells. So inside your cells, iron is stored in the protein called ferritin. And like I said before, in blood, iron is stored in the protein called hemoglobin. And unfortunately for us, Listeria have systems to take up all these different types of proteins and use this iron for themselves. So not only can Listeria steal iron from our bodies, but it also can steal iron from other bacteria that are normally found in your gut. For example, E. coli produces a protein called siderophores, which are released into your gut to bind iron, and Listeria can use these siderophores for themselves. So it looks like here that Listeria has the upper hand in terms of acquiring iron inside the body. So how Listeria controls iron uptake? So all living things possess genes, and genes are the basic units of DNA that are the building blocks for your cells, and they allow traits to be passed down from your parents to offspring. So genes control things like eye color and hair color. So Listeria has specific genes for iron uptake. And what we did was we used a computer to identify several genes in Listeria involved in iron uptake, including the gene LMO0641. So where my work began, we wanted to make a mutant strain of Listeria in which I mean we deleted this LMO0641 gene, which makes iron available to Listeria. So we have the wild-type Listeria that possesses LMO0641, which allows iron to be made available to Listeria, and we have a mutant strain in which we've deleted the iron gene. So what we wanted to do was compare this wild type and the mutant strain in conditions such as low iron and infection. So normally when growing Listeria, we grow it in a nutrient media called BHI, and Listeria can either be grown on solid agar or liquid broth. So before we started our experiments, we wanted to make sure that in normal nutrient media, there was no differences between the wild type and this mutant in terms of growth. So as you can see here, this is just growth over a certain amount of time, and there was no difference between the wild type and mutant, which is perfect. But where we did start to see differences between the wild type and the mutant strain was growth on low iron agar. So if you look at the plate setup, WT stands for the wild type, which has the iron gene. The triangle represents the mutant, in which that gene is deleted. And C stands for the complement, which is the mutant strain of Listeria, where we put back in the iron gene. So the iron source in this experiment was ferric citrate, and these are just plates with increasing amounts of iron. So as you can see, the first two plates, no iron and 2 millimolar ferric citrate. The iron is too low for any of the strains to grow. But when you increase the iron amounts to 3 and 4 millimolar ferric citrate, both the wild type and complement that have the iron gene are growing fine, whereas the mutant in this low iron environment is not able to grow. When you increase the iron in the media, again to a higher concentration of 6 millimolar, the growth of the mutant is restored. So similarly, on low agar, the same thing was seen in low iron broth. So at 0.25 millimolar ferric citrate, this mutant displayed a growth defect compared to both the wild type and complement. But when you increase the amounts of iron in the media, this growth was restored. So the big question is, can this 0641 mutant cause infection? So we know bacteria need iron inside the body to cause infection, and we've deleted a gene that makes iron available to Listeria. So if you look at this mouse model, after 3 days, you can see this mutant is not able to cause infection as well as both the wild type and complement. So this graph just represents the numbers of bacteria that are able to survive in the spleen after 3 days. So as you can see here, because this mutant is not able to cause infection as well, this gene is playing a big role in terms of infection. So what we know so far is we've identified a gene that makes iron available to Listeria, and this gene is responsible for growth in low iron as well as full infection. So what's the relevance of this? It's possibility that we can use this 0641 mutant as a vaccine. And a vaccine is just a biological preparation of either weakened or inactive microbes in order to initiate an immune response. So basically, here's a few different types of bacteria that cause illnesses that we all know, such as strep throat and pneumonia. And just like we each have our own unique fingerprints, these bacteria have their own unique cell surface proteins that are unique to that bacteria. So when our body makes an immune response, we have immune cells that recognize these specific cell surface proteins, send out immune cells to kill these bacteria, and we remember them for when next time we get infected, we can make a better, faster immune response. So similar to bacteria, viruses such as HIV, influenza, and the human papillomavirus, which can cause cervical cancer, have their own specific cell surface proteins. And it's our hopes one day to be able to use this Listeria 0641 mutant as a possible vaccine delivery vector. And by that, I mean express these cell surface proteins from other viruses on our mutants and use it as a possible vaccine.
A minute to go.
So the reason this would make it, which is what I'm currently working on now. So the reason this mutant would make a good vaccine is because it survives inside the body, it does not cause infection, we can present these cell surface proteins of other viruses to our immune cells, and we can allow our body to make an immune response either to the viruses they cause or the tumor cells they cause. So just to sum up, this is just the pathogenic cycle of Listeria, and Listeria can move freely and continuously from the environment to food to the body. So by understanding how Listeria causes infection, we have a better chance of controlling it and ultimately preventing it, in hopes of one day being able to break this cycle. So thank you very much for listening.
Just to clarify, when you talk about developing a vaccine, are you talking about using the mutant to as a vehicle for delivery of vaccines against other viruses rather than a bacteria?
The bacteria itself. Yeah, recently, so far in my work, I've shown that this bacteria can survive inside the cells and it doesn't cause infection. But last week, we actually showed that we did an initial vaccine trial in which we showed that this mutant does protect. When we give mice a booster with this mutant, it actually does protect against the wild type infectious strain after so many days. So it can be used as a vaccine against itself because Listeria, like I was saying, like any other bacteria, it produces its own specific cell surface proteins, and so not only would it be able to make an immune response to itself, but also we could use it as a vehicle for delivery of other cell surface proteins as well.
Just to clarify, was the identification of this mutant, was that original work here? Or
it was original work here. It was actually the person who did her PhD before me. She identified the gene, and then I went on to work with it.
So, what percentage of listeriosis would you say is diagnosed or is detected because the symptoms in those who are not immunocompromised or would sort of, most people would experience those on a regular basis?
Yeah, I'd say it's probably, because it is undiagnosed, there probably is a little bit higher of a percentage because most of us are able to clear the infection and we just maybe would feel like we had the flu. So most people in those circumstances wouldn't bother going to the hospital. But I still, in terms of actual numbers that our people are affected, I don't think it's, in comparison to the numbers of other foodborne bacteria, like I said, like E. coli or Salmonella. But that's another reason that it's a good candidate to be used as a vaccine, because I don't think most people would have already had an initial immune response to Listeria. I think most of the population wouldn't have had the opportunity to experience, Listeria. So I think it would be able to initiate a good immune response because most people haven't seen it before.
I'm interested in you saying that it comes from animals through to humans and through the earth and that. And what proportions in terms of, what movement is it from the land?
Some people speculate that soil is a natural reservoir for Listeria, but we don't know that for sure. But we do know that it is present in the environment. Whether that's its natural habitat or not is still yet to be seen. But a lot of times the contamination happens at the food industry bit of it. So even let's say there's a lot of small farmers in Ireland that go on to produce dairy products that we all consume, and just something as little as not separating the farm area from the actual production of the cheese can actually lead to the contamination. For instance, just like walking in the soil and you might get Listeria on your boot or on your coat, and then you go into the production line. That's where a lot of the contamination of foods happens, or not cleaning or sterilizing equipment properly. So I think a lot of times in terms of Listeria and other foodborne diseases, prevention at the food industry part of it would eradicate a lot of the diseases that we see.
Thank you very much, Heather.